give a little bit of a forum for the European projects to introduce themselves into the field. And obviously we also want to present a forum for communicating and discussing a number of societal issues, public perception, ethics, IPR. It's not quite clear how these sessions or the respective sessions are going to relate to what we have here today, because honestly many of the things that have matured very much here in the US have not matured so much in Europe. We're just pretty much starting to get our act together. And we have a lot of open questions here in this area, but there will be definitely also a part of the conference time devoted to these topics. We have assembled a scientific organizing board that we hope is going to help us to select a list of high quality presentations for the event. And all I can say is we look forward to see many, many of you in Zurich a year from now. You're welcome.
Next up is Randy Rep Berg from MIT, who's going to be talking about iGEM, which has been just a fantastic way of getting young people excited about synthetic biology. And we'll also be talking about the registry of biological parts in 10 minutes. So that's right. Take it away, Randy.
Okay, so now we have to talk very fast because we only have 10 minutes. We have 2 topics to cover. I started off in the 30-year career in the computer industry. And then I came and joined my friend Tom Knight at MIT to become a biologist. That was fundamentally an impossible task. You can't be a 30-year career person in computing and then become a biologist. You have to have gone through the entire PhD program in biology to do that. So the only solution was for all of us to make a new field. And so we've been working on this field of synthetic biology so we can have jobs. So let me make this go for a sec. Okay, it's looking good. Okay, so there are 2 URLs you need to know. The pattern is easy, parts.mit.edu, and then it's /igem or /registry. There's no www at the front. When I got to MIT and started to do some of the basics of biology, I found kind of a basic truth, which is that the life of a biologist is difficult. What you do is you struggle, you have some success, you get to write a paper, you get to go to a conference, you go back and you struggle. This is crazy, right? No, what you want is you want a better life. You want struggle, that's good for your character. Then you want success, and then you want success more and more and more. That's what the computer industry did, right? It's not like you figure out how to make a processor chip, you struggle, and then you got to go start all over. You build on everything you've had before. So that goal of can you build on what you have previously done in biology is kind of summed up by this idea of parts. Can you make biological systems from standard and interchangeable parts and operate them in living cells? A lot of the biologists would say, no, you can't. Biology is just so complex that each case is unique. It's a hopeless task. We view this as not a scientific question, so the scientists might not be right, but rather as an engineering question. So we have to figure out what to do about that. Now, we laid out the abstraction hierarchy, and Drew talked about that in some detail. But if you think about what's happened over the past 2 days, we've had lots of presentations by lots of people who are showing a discovery or a demonstration of something happening in biology, right? And we're excited about each one of the presentations we see. They look very exciting. I'm looking beyond that and assuming that all of those things are going to be made to work. That we will, in fact, make parts that are interchangeable, that will make parts that have good behavior, that behave well inside the cell. And when all of that happens, the world will be different than it is now. It will be possible to have an industry of synthetic biology. And this will be an industry revolution, like the electronics industry, the software industry, the network revolution, maybe even like the industrial revolution. If we're going to do that, if we're going to have an engineering discipline that's based on parts, we're going to have 2 more things. We're going to have catalogs of those parts, so you know what you buy. And we're going to have suppliers of those parts. So we started with The first version of the parts catalog, this is the new version. If people are used to seeing one with a big strong border on the left, this is the new one. It will be released on June 3rd, and everybody can make use of it. It will actually be a lot more fun. In this part catalog, we have a lot of different kind of parts in categories. So here, for example, are a number of signaling parts that are used to do signaling from one cell to another. They use small molecules that go in the media between. These parts come in signal senders. A bunch of signal senders, signal receivers, other signaling parts, and the list of other signaling parts goes on for quite some time. Each of the parts has a description. Each of the parts has a description that talks about what it does in kind of a user, human understandable way, talks about its performance and properties, and then provides a specification of how it is designed and what it's made out of. We have characterized some of the parts. This is like my best table, right? These are transcriptional terminators, where they are supposed to terminate. So efficiency, which is how good a job they do of terminating, ought to be like, 99 or 100%. One might note that a lot of them aren't really very good, like 0.3%, which means they're more like a resistor. They leak a lot. This one actually is 0.3 and minus 0.3. That's because it's a promoter in the other direction. So the parts actually have a lot of trouble, but some of them are pretty good, and a bunch of them really work pretty reliably. We have a repository. I think it's like the gold repository, which are the parts in DNA in our freezers that we send out to all of the schools that participate in iGEM and we make available to members of the community. At the end of last year, the registry had about 88 promoters, 130 basic protein coding regions, 30 reporters, some RNA parts, transcriptional terminators, and lots of project parts. About 702 parts were available as DNA. Lots of parts have been specified by the teams over the summer, but never implemented as DNA. Finishing up on the registry. The registry that will be released in June is now a blend of a wiki. Does everybody know about what wikis are? It's something to go learn about. They let you edit and change the content of a website as a community. And we're bringing that technology into the parts registry while at the same time preserving the database aspects of the registry. So we have both the wiki content and the hard content. The thing that we're working on after this summer is a question of how to arrange the registry so that we encourage a broad community of people working on parts. The answer to that, in my opinion, is to not have a centralized registry, but rather to make the protocols and technologies using some of the semantic web-like things to allow many universities, industrial firms, governmental organizations to all have their own registry in a similar way to the semiconductor industry where lots of vendors offer their own catalogs of parts. They get to represent their parts in the way they like. Okay, so if you think about that engineering question again, we want to know, can you build biological systems from standard interchangeable parts and operate them in living cells? How are we going to find out? The answer is that we at MIT have a resource. That resource is students. So what we do is we find undergraduate students. Undergraduate students don't know very much about what you can and can't do. We don't tell them, and we let them go ahead and build things. This was done first during the January independent activity period at MIT in 2003. This was thought up by Tom Knight, Drew Endy, Jerry Sussman, and I, and we decided we would just try it out and see how it worked. We got 16 students. They divided into 4 teams. We said, you have a month to design systems. We have money to synthesize the DNA. Go forth and design. That worked very well, but January was very short. We did it again the following year, 2004. One of the projects done by a 5-student team was the Equilibrator. This was a project to make a polka dot. They decided they would make a polka dot not through some gradient process, but rather by having cells swim together into a clump. They made a animation of their project. They made a system-level diagram of their project. They did a population simulation to show how good a polka dot it would make. And they laid it out, their entire system design, as parts. January was over. That summer, we found 4 other schools who were similarly optimistic and were our friends and wouldn't complain too much if things didn't work out. They were BU, Caltech, Princeton, and UT Austin. Each of those schools put together a student team, primarily undergraduates. We got a large DNA synthesis budget. We had money from NSF and some from DARPA. And we held a summer activity to do engineering of biology, and then a jamboree in November. These are the students. You can see how happy they are, right? They had a good time over the summer. We continued this on in 2005. We had 13 schools. There's the list. We went international. We had ETH in Zurich. We had Cambridge, UK. They call that Old Cambridge. And then we also had Toronto. So this project was basically an expansion. We did things better than we did last time in 2004. Worked out pretty well. Here are a couple very quick versions of the projects done by a couple of those schools. The first is University of Cambridge. I have taken out all the animations in a lot of the slides. You can see they have great graphics. They were working on chemotaxis in E. coli. They developed a very complex bacterial traffic light system with a lot of switches and things and lights. They added parts to the registry. They got the chemotaxis working under control. They did a flipping DNA switch and a few other things, and then the summer ended. You can see this is their team. Again, very happy, not a very big team. The other group that I'd like to highlight from Europe was ETH Zurich. They tried to make a system that would count. They developed a system design. They made a piece of it work, actually. They made models for their systems. And one might note that here we've labeled green or black as to whether the students were biology or not biology, and it was a very mixed group, quite successful. Even a tiny college, Davidson College, was able to have a team that was able to have a team over the summer. And they looked into fluorescent proteins quite a lot and did quite well with that. So that was 2005. It's now 2006. We've grown again. There are 40 schools this year. We're much more international. We have a school in India. We have 2 in Japan. We might have 1 in South Korea. We have 3 in Canada. We have 1 in Mexico City. We have a Latin America team, an African team. We have 3 in the UK, Turkey, and 4 more in continental Europe. And then there are a whole lot of the schools in the US, the Harvard and Caltech, and lots and lots. So go to parsons.mit.edu/igem. You can see the list of the participating schools, and you can see website or wiki information about what many of the schools are doing. We will again have a jamboree in November. So if you're interested, come to MIT first weekend in November. We have ambassadors this year. 2 of our ambassadors are here today, right back there. They are responsible for helping the schools be more successful this year. And so I think that's going to work out and they'll do a good job. One of the things that's happened, by the way, is the EU has provided some funding for it this year. They provided 50,000 euros, which has been divided up between 6 schools to help pay for the participation in iGEM. iGEM 2007, for the first time, I have to start working on that now. I'm expecting and preparing for order of 100 to 120 schools. Typically at a school, they'll have somewhere in the 6 to 12 students, a number of instructors and faculty members advising. One of the things that has happened that's very surprising, obviously the students are going to have a great time, but the instructors have also had a great time, and they have then moved synthetic biology into the curriculum at their school. In fact, this year, quite a few of the schools are already teaching courses on synthetic biology in the semester before the summer to get their students ready for the competition. So it's become a pretty good thing. The final thing I'd like to just end with is that in thinking about all this issue of risks and bioethics and all of that, there's a curve. I don't know what the shape is, but I know there are things on the curve. There's like basic happy researchers. One would hope that they're constructive and that there's a lot of them, right? We have, garage biohackers. We hope there's not as many of them. We hope they're still pretty constructive. We hope we don't have very many disgruntled researchers, and we don't let them back in. And over here we know we have Ben Laden Genetics Inc., and they work on the cure for cancer during the day, right? it's hard to do anything about that. But we need to make this really bloom. Thank you.
Tom Knight, also from MIT, we've got real diversity on this panel, is with the AI Lab, and I know first introduced me to some of the ideas around synthetic biology in a DARPA ISAT study that probably over 10 years ago on a virus that was going to make computing.
Just about 10 years ago. So it's a real pleasure to be here. We've heard a lot about standards today, and we heard a lot about standards for the past several days. I'd like to sort of talk this afternoon a little bit about where we are with standards and where we need to be and where we're not, and to try to solicit your help in creating the next set of standards that we are all going to need in this technology. Drew showed a quote from a picture of the paper by William Sellers. Sellers in 1864 developed the first standard for screw threads in the United States. We all take it for granted that our screws have standard sizes. We all take it for granted pretty much that they're all right-handed screws. Where did that all come from? It didn't all happen miraculously. Somebody actually sat down and said, this is the way we're going to go do it. And in the United States, it was Sellers. In the UK, it was Whitworth, who actually did it quite a few years before Sellers. So we really are in a similar sort of situation right now where everything that we're doing is I guess, in the scientific community, largely handcrafted. The plasmids are all lovingly tended and put in your freezer. Every one of them is different. Every one of the restriction sites is different. Every time you want to do a manipulation on a piece of DNA, as I explain it, you want to do an experiment, it turns into two experiments. One is the experiment you wanted to do, and the other one is the experiment in making the piece of DNA that you want to use for that experiment. In the engineering world, we have a tendency to try to lay down these standards, and the standards have several roles. One of them is they allow us to simplify the way that we think about the interfaces between components. They provide that interface, and they provide a way of hiding the complexity at the lower levels, allow us to think about composition of components both at the structural level, can you put them together and hold them together, and at the functional level. If I add, put one inverter into output into the input of another inverter, then I'm going to get a system that works. They also provide a way of writing contracts for commercial use. So when I type a sequence into a web browser for an oligo, everybody agrees what the A, G, C's, and T's are. That's not a given. Somebody decided what to call, how to indicate that sequence. And we need similar sort of mechanism for precisely specifying these interfaces, these societal interfaces between groups that are doing different parts of the same job. They also allow independent evolution of different parts by different people. So Randy talked just a minute ago about the idea that we could have different manufacturers for parts. And that only works if you have a standard so that the parts that are put together from Texas Instrument work together with the parts from AMD. And usually that works in the electronics industry. We need to get the standards in place so that that will work in the biology community that we're looking at now. The standards also, in addition to constructing the systems, we need the standards that are going to allow us to measure and test these systems in a form that is comparable, so that the results from Stanford and the results from MIT are when we measure a system are going to mean the same thing. We're going to measure in the same units. And so there's this joke that the good thing about standards is so many to choose from. Unfortunately, in this field, that isn't really true yet. So, we really need some more standards. And I'm going to try to pin down some of the standards that we are going to need. So this is a pass at some of the areas I think that we either have now or are going to need very desperately some standards in the emerging field of synthetic biology. We already have a few standards in the area of structural DNA assembly. Some of those are, and I'll spend some time talking more about this in a minute. Some of those are working right now. We may need others. There's, a couple of competing standards, and probably, this will continue to evolve, but it's one of the places where, at the moment, things are under the best control, probably. I mentioned before that, it's not enough to be able to weld the pieces together. You need to be able to also interface them and have them function together. We are much further away from having good standards in the functional area than we are in the assembly area. And again, I'll talk more about that in a minute. We are practically nowhere on the measurements issue. The measurement of the properties of the devices and parts that we're putting together are in a very primitive state. We desperately need help on that, and I'm very anxious to solicit the help of the people in the audience and suggestions for how we can push the technology for part measurement. The standards don't necessarily end with the biological parts themselves, but I think encompass some of the things that Randy was talking about a minute ago. We want standards for the documentation of the parts. We want standards for how you publish about these parts. We want standards for what those data sheets look like for the parts. We also need to start thinking about how we document some of these issues that arose this morning. What are the safety properties of these parts? How are we going to quantify how dangerous a particular part is? Or is it only dangerous in combination with some other part? And how do we document those issues? We also heard some discussion, I think, early this morning about the idea that we might be able to intentionally tag some of the constructs that we made in the biological world. So when you make an artificial construct, maybe the idea is you intentionally put together, put into that system an identifier that says, Tom Knight made this, come see me if there's a problem. So in the structural area, as I said, we're in pretty good shape. We have an existing standard that we've developed in our laboratory, which is this BioBrick standard. I don't think anyone thinks it's perfect. It is working for us. It is working for some of the people that we're collaborating with. This is the third version of that standard, although nobody else saw the first two, thank goodness. Austin Chi, my student, came up with a much more sophisticated version, which we actually haven't been pushing, which actually has a lot of good features, and so that might be deserving of some further attention. Pam Silver's lab, particularly Ira Phillips, has developed a technique for doing protein domain fusions in the assembly process. And of course, total synthesis may replace a lot of these assembly issues, as we've talked about yesterday. In the system insertion, you want to be able to take those pieces of DNA and put them into a living context. Here we have other issues. We have issues of what plasmid do you put these into? We have issues about what organism you put them into. Do we put them into E. coli or into Invitrogen top 10? Do we put them into E. coli even, or some other bacteria like Subtilis or Salmonella? We heard of some very compelling reasons for using Salmonella. Are there other substrates that we want to think about? Do we want to think about multicellular ecologies of systems that we put together? So the system insertion issues and the standards there are very important. On the functional compositions end of things, we do have some standards in place right now for connecting transcriptional control from one place to another, but The whole issue, which came out earlier, in the conference of, I think Samantha Sutton's work in particular, of getting control over the post-translational logic of phosphorylation, for example, of one protein by another, and sending signals from one part of the system to another in those forms. We have no standards whatsoever in that space. So we have a set of standard inputs which we need to characterize and specify. We have a set of standard outputs which we need to specify and control in these systems. Both of those are ripe for control. In the measurement area, there's hundreds of things that we want to measure that we can't measure right now. Here's a partial list. There's a lot that, essentially everything that you want to know scientifically about the cell, we would like to, from an engineering standpoint, also be able to measure and quantify. And the quantification in the engineering world tends to be at least as important, perhaps more important than in the rest of the scientific community. So Randy already showed you about the Registry of Standard Biological Parts. I'd remind you all also of another website at MIT which is very, very important to us, which is the openwetware.org website. This is a laboratory-wide web, multi-laboratory across the world kind of website, collaborative website, which allows researchers at any lab to join the Open Wetware group, to document their protocols, and it's an emerging, I think, as a very, very important resource certainly for my research. And I'd encourage you all to at least take a look at this website and, ideally contribute and start to become part of that community. And I think I'll leave it at that, unless there's some questions. And, and if anybody has ideas on good standards, I am interested in hearing about it.
True.
This is going to be real quick. So here's that website Tom was just looking at, but now a different page. This is the Synthetic Society page referred to earlier. One of the things that's come up as a point of conversation is whether or not we might think of ways to best organize ourselves in support of doing all of the stuff we need to get done. Right, so we have this wonderful conference series, but something else perhaps. So scroll all the way down to the bottom. There's the beginnings of a section on community organization and culture, and you might think that Tom and I spend our evenings looking for esoteric photographs from the 1800s, but we really spend all of our time doing this. What I'm showing here is the picture from 1855 of the third annual meeting of the American Society of Civil Engineers. And so we're doing a lot of engineering in the context of biology, and I don't know of the American Society of Genetic Engineers or the International Union of Synthetic Biologists. I don't know if there should be something like this, but what I'd like to do then is simply point out that stuff like this happens, and in the context of trying to get all the things done that we need to get done and having better focusing of resources and points of accountability, if we actually wanted to implement things like professional codes of conduct, how would we actually do that? On what time scale, if ever, do we need to think about a more formal sort of organization?
Okay, so Drew definitely wins the prize for brevity. And I still remember that my high school English teacher giving me the quote of the person who said, "I'm sorry this letter is so long, but I did not have time to make it shorter." So we appreciate that, Drew. So I'm not only your moderator today, but I'm actually going to be giving a presentation that really is based not so much on the work that I'm doing today at UC Berkeley, but from my checkered past. From 1993 to 2001, I was in the White House National Economic Council working for President Clinton and Vice President Gore, as we say, during those dark days of peace and prosperity. And And one of the things that, if you guys put together conferences and, your sort of unit of work is the publication, for someone that was in my job, namely working on issues related to science, technology, and innovation, we thought in terms of initiatives, that is, new ideas that the president and vice president were going to get behind. So what I wanted to do is to ask a series of questions about what stance this community might take with respect to federal funding for synthetic biology. And the first thing that I wanted to point out is that funding decisions, and I'm sorry for the Europeans in the room, I'm going to be parochial and talk about the US, but research funding decisions get made at many different levels in Washington. And I'll just give you a couple of examples. Starting around 97, 98, there was a successful campaign to double the NIH budget from $14 billion to about $28 billion. And that came about largely because you had a number of the disease groups who got together and said, rather than forming a circle and firing inwards about what is our relative share of the NIH pie, why don't we all agree that the NIH budget should be doubled? And they organized a successful campaign to actually do that, that was supported by the Congress. Now, you might think that was a good idea or a bad idea, but that's one example. The thing that I'm going to be talking about is the process of developing national science and technology initiatives. I worked on three during the Clinton administration. One was called the Next Generation Internet Initiative, which involved a collaboration with a number of research universities and is still going on with an organization called Internet 2, which has developed a backbone which is, I think, up to 10 gigabits per second at this point. The National Nanotechnology Initiative, which has resulted in funding for nanoscale science and engineering going from $270 million when we started to over $1 billion today, and has continued to enjoy support during the current administration. You can have agency-level initiatives like the NSF effort in cyber infrastructure, or a DARPA program manager can come up with a new idea, and if he or she is successful in convincing the director, have a new program. And then you also have the cumulative impact of many individual investigator decisions about what type of research grants to submit and what actually make it through the peer review process. What I'm going to be talking about today is primarily this idea of a national science and technology initiative, which involves getting multiple federal agencies together to decide, hey, this is really an important area, this is an area that we're going to invest more money in. So the synthetic biology community could have a number of different viewpoints. They could treat federal policy as a given. Washington is a black box. We don't understand it. We don't understand how to influence it. And just how do we figure out how to optimize within that? Or the community could say, how do we make sure that synthetic biology plays an appropriate role in other national research initiatives and priorities that are either ongoing right now or are in the process of formation? Here, clean energy and nanotechnology come to mind. You could make the case for a separate initiative specifically on synthetic biology, or you could make the case for a broader initiative that would be include but not be limited to synthetic biology, like a national advanced biotech initiative or an initiative aimed at enabling the bioeconomy. And I think one case you could make is that currently in the United States, funding for biology, since it's dominated by NIH, really does not pay a lot of attention to non-health applications of biology. So a vision around enabling 21st century bioeconomy and trying to figure out what research would need to happen. And synthetic biology would be a component of that, but would not necessarily be the only element. So if someone came up to me and said, you have 48 hours to come up with an initiative in synthetic biology, on the basis of listening to this conference, I think these are some of the potential elements. I think a strong case has been made that we need a lot more work that needs to happen in foundational technologies, the notion of parts, devices, and systems that we've heard about, applications of synthetic biology, and I'm going to give you a list based on just what I've heard from this conference, a requirement to expand the education and workforce in this area, building on programs like iGEM, but you could also imagine training grants, course development, a lot of infrastructure and tool development that needs to happen, things like parts devices, and clearly work on the ethical, legal, and societal implications, and both understanding and mitigating the risks. And I also threw in prizes because I think that's a really interesting way of stimulating technological innovation, because it allows the government to set an objective without specifying the best means to reach that objective. So in terms of applications, we've heard about carbon neutral energy, health, tumor-eating bacteria, synthetic biology as a tool for basic biology and biomedical research, information processing, environmental remediation, synthetic biology playing a constructive role in the area of biodefense, carbon sequestration, focusing on closed loop as opposed to environmental release, green production of bulk chemicals, nanoscale fabrication, and applications of synthetic biology for the developing world, such as Jay's work on reducing the cost of making the most effective anti-malarial drug by a factor of 10. So I think you could make a very strong case on the base of the breadth and potential impact of these applications that one should have a national initiative in this area would be worthwhile, and that this is too broad to fall under the province of any one particular agency, but that one would need to build a coalition of federal research agencies to support this work. So what would be the benefits of such an initiative? I think that obviously increased funding would accelerate progress in the field and allow us to achieve some of the benefits of synthetic biology. You could have a more coherent investment strategy. It would certainly make interdisciplinary collaboration easier. It would attract more young researchers to the field, and it would also catalyze greater investments by states, universities, incumbents, startups, and foreign governments. One of the things that certainly happened after President Clinton talked about the National Nanotechnology Initiative at a speech he gave in Caltech, and then also talked about it in the State of the Union, is that the next year every state decided that they needed to be making investments so that they could be the nanotechnology valley. I think there were over 30 foreign governments that followed the United States in also investing in national nanotechnology initiatives. You saw a lot of increased startup activity and activity by Fortune 500 companies. Universities started allocating faculty positions to this areas and going out and raising money for more shared facilities. So clearly there is some value to increasing the awareness and visibility of a particular area. However, I also think that there are some risks associated with this. There's also the potential for hype and backlash. That is, the research community can overpromise and underdeliver. And Tom can certainly tell you about AI winter, which is a phenomenon that happened in the 1980s. You might decide that national and policy attention may be premature. Maybe the field needs to get its act together a little bit more before pursuing a national initiative. A lot of what happens, and this certainly happened in the area of nano, is you have relabeling of existing activity. Initiatives are always harder to sustain than they are to launch. So Washington suffers from ADD. And a lot of times, particularly if these initiatives are politically driven rather than being driven by the community, they can be overly prescriptive. So you have the phenomena of Washington saying, rather than Washington saying, we need carbon neutral energy, we need to improve energy security, saying the answer is a hydrogen car, and we're going to fund research to do that. And, you know, that may work out, but a lot of smart people believe that there are a huge number of scientific and technological and economic obstacles to doing that. And, you know, defining the answer as opposed to posing the question and letting the best ideas bubble up from the research community can be problematic. So in terms of what homework needs to be done by a research community to develop a national science and technology initiative, you need to have ambitious but plausible goals that capture the public imagination and respond to national needs. You have to identify what are the most promising and important research topics, identify what you think are the right modalities of funding, both in terms of the right mix of performers, the scale of grants, the balance between peer review versus tradition in DARPA of having a strong program manager that can really put together a portfolio of projects that he or she thinks are important. Think about what's the capacity of the existing community to spend increases wisely. What would you prioritize at different funding levels? What's the right division of funding, division of labor between the funding agencies based on their mission and the resources that they have? And as we've been talking about today, what are some of the policy issues that go well beyond R&D in terms of things like biosecurity, IP, ethics, and regulatory approval? So based on my own experience of putting together national research initiatives like the National Nanotechnology Initiative and NGI and increased funding for long-term high-risk IT research, these are a couple of the things that I think the community would need to do to increase the chances that there would actually be a national research initiative either in synthetic biology or in a broader, more encompassing category, which I actually think might be wiser, that would help get these issues on the national agenda. Thank you. Okay, so the next thing we're going to do is have some open discussion. So I would invite all of the participants in the panel to go up and sit in the dais, and I will moderate questions from the floor. Oliver.
Actually, Tom, I just wanted to confuse things by asking a question to you specifically. How much do you worry, especially if we were thinking that one of the things that a governmental program might do would be build the thing we heard about this morning, which is a synthesis farm on the scale of a contemporary sequencing farm? Do you know how much you worry about that crowding out the private capital that's gone into synthesis companies? And isn't one of the obvious things for a government initiative to do to go in big and heavily capitalized on actually doing a lot of synthesis and thus possibly damaging the incipient industry that's been here today?
Yeah, no, I mean, certainly one of the questions that you need to answer when putting together a research initiative is to make the case for federal involvement. And the question that will always come up is, you know, if this is an area that eventually has economic and commercial potential, why don't we just leave it to the private sector? And so I think that the answer that is traditionally made is that, particularly if you're talking about longer term and higher risk research, companies cannot appropriate all the benefits from that. So I think the government is on far stronger ground when it identifies an area of research which is beyond the time horizons of most companies. And so if there is a thriving DNA synthesis industry, then the role of the government might be to improve the purchasing power of the research community as opposed to try to recreate that capability in a national lab.
Tom, I had a question. I was wondering what the broader agenda you would recommend that we try funding rather than synthetic biology. What would be the nature of that broader agenda?
Well, I can't tell you what it is, but I can tell you what it would be motivated by, which is a sense that biology is going to have an impact that goes far beyond health. and that if you look at current support for biology, over 80% of it is done by NIH. And so I think there's a lot of important areas where in sort of non-health applications of biology, that there's probably more investment that could be made. So I think that might be a central organizing principle, that is, enabling the bioeconomy. But in terms of, what are the specific research topics that would go under that, I think this community and other communities in biology would be the ones to identify that.
So just to follow up on what you were just saying, Tom, I think perhaps the flip side of, instead of trying to go for something that's not health, because most of it is health,
right,
is to actually go where most of the investment is, and that there's a lot of synergy, and there's a real nexus in the Congress and other places for this is an important thing to fund.
Yeah.
So especially in the context of sort of the risks that we've been talking about, the risks of bioterrorism, of nasty things happening to people in a health perspective, actually being proactive and saying, we're going to focus on, I don't know, eliminating infectious disease. I mean, you can pick your title, where you actually then could really jump onto already pre-existing sort of motivations, let's say, within decision making to do that, as opposed to trying to create your own new bioeconomy, or trying to gin something up from almost whole cloth. I mean, there's different ways to do it, obviously, but I think that's just a different perspective.
No, absolutely.
This is about the public profile and the media strategy of the community. If somebody writes something nasty about you in a newspaper, it's a debate within yourself. Do you respond so that the journalists have something on the record and in the cuttings files, or do you just let it go? So you had an open letter published, which is being picked up by the media, which has amongst its signatories powerful organizations that in certain circumstances can have traction over and above the heads of the scientific community. And it contains within it an argument about self-regulation or public regulation and law. It also has, in the background notes, which weren't circulated, some specific allegations about the risks and the fears that surround this technology, which were sort of explained, put in a context in the ethical discussion we had today, but nevertheless absolutely specific to synthetic biology. And the question is, do you respond or not? Do you as a community decide, even in the absence of a learned society that could have a media representative and communication, do you respond? Do you actually do one of two things? First, do you have a briefing at the press club to ensure that the journalists at least hear what your perspective is on your community and your activities? Or do you even prepare a point-by-point response to that open letter?
Drew, do you want to take a crack at that?
Well, I'll jump in for a sec while Drew figures out what the right answer is. I can't imagine the right thing is to do a response, because what that's going to produce is the press completely covering the conflict, not covering the commonality, not covering the issues, but he said this, he said this.
Yeah. The only problem with that theory is, that was Michael Dukakis' theory in 1988.
It worked out pretty well.
Which is that if I don't respond to Bush's attacks on me, it won't make news. And unfortunately it did. So I have the scars on my body to. So I don't think, I'm not sure that, ignoring it and hoping it will go away is the answer.
No, but I was only saying that responding directly, kind of point by point. Yeah. On the other hand, the right answer is probably to, for the most part, be ahead of the topic.
We've got a really cool field, and maybe microphones that work sometimes too. Do I have to do something here? Is this working now? Right. So we've got this cool field called synthetic biology. We've been paying approximately no attention to our interactions with people outside of the community. We get to hear talks, as was mentioned earlier, with words in them like gene killer, creating life, and so on. And part of me feels that it's appropriate to use these sorts of words in the language in general, because there are issues associated with the development of this technology. And as has come up in other forums, it would be inappropriate to rename the field shiny, happy biology. I think the specific case that you're bringing up in response to the release that came out on Thursday in front of this meeting is an unfortunate result from some miscommunication regarding what was actually going to happen today. Having said that, there are also issues raised by those organizations that aren't necessarily topics that we as a community are addressing, and I'm not sure what the right response to those would be. I love the fact that one of the strengths of everybody in the room is we're more or less completely forthcoming with both what we're doing and our understanding of the issues, and I wouldn't want to see that go away. The full range of advice has already been discussed in terms of what the appropriate response would be. Where we're, so on the one end of the spectrum, it's do nothing, do not acknowledge anybody besides ourselves. You're just giving voice to people who would otherwise, do nothing but get more attention, versus you've got to be purposefully inclusive and bring folks in and see if you can make something of it. We'll figure it out, right, over the immediate future as we put together the declaration. I think it'll certainly include an explanation for what actually happened and some stated hopes for how things might play out over the next 6 to 12 months as we head into Zurich. And if anybody has specific, the actual right answer, please help everybody else figure it out.
Lori.
I don't know if it's the right answer, but I can say something about timing, which is that this is the time for a lot of talk, right, between now and, say, a year from now, to really listen to those calls for inclusivity and discourse and how you're going to do it. It's interesting that the NIH strategy for the doubling of the budget not only took John Porter, senator from Illinois, I might add, to really be behind it, and a few senators to get it. But it took a constituency. It took people saying, I want this. It was the disease groupings that made the decision to go forward. And I think that that's a good way that democracy works. That why Congress gives federal money, why I pay Congress to give you federal money, is because you want it to be responsive to the concerns and needs of citizens. So there is a potential coalition that could be built, but people who care about something like healing and health, but the broader issue of the brokenness of the world. Global warming is one clear problem that people are now coalescing about. The environmental concerns that are linked to that. And I think that's one of the extraordinary reasons why the field, as Drew says, is so cool. It's not just cool because it's interesting and complex and a brilliantly interesting way to reconfigure the world. It's interesting because people see in it the hope for that repair in the way that people saw in regular biology or genetic engineering or the war on cancer, whatever, the hope for health. So that the inclusion of the disparate voices, people who query it, people who are concerned about, people who yearn for it, into the kind of constituency that would then support greater research funding for it is, I think, one of the goals that you might think about. And it's, I think, the real response to the letter that was circulated, to reflect on what was said and to then think about the other groups in the larger world that surrounds this interesting community that need to have a voice, patient groups, environmental groups that might yearn for this. So I think that's it, to broaden that, actually, rather than fleeing from it, to actually broaden that and include more voices.
So Drew raised the question of a more organized society, which I would be very in favor of. I was curious, though, if people had counter-arguments against that, against having a more structured professional society of synthetic biology of some form. And in particular, is there an impediment to organizing, if we chose to go ahead with it, is there an impediment to organizing quickly versus, say, making a decision like that a year from now at the next meeting when we have our next, as much of a critical mass as we're going to have of synthetic biologists?
I was just hoping to, plant a seed for discussion, and since you're bringing it up, I don't know.
So do you think it might be too premature simply because we haven't had enough time to discuss it yet? Maybe?
Yeah, by definition, yeah. Right, so something to figure out.
We don't even know who is a synthetic biologist and who is not. We can't decide for ourselves what the field is yet. So it would be probably a little premature to try to decide who's in and who isn't.
Well, I definitely see that point. On the other hand, forming something a little bit more organized might be a better way to pull those people out of the woodwork. I don't know if there can be an intermediate body of some form.
Okay. So unless there are any more questions, we're going to wrap this session up, and we're going to have Jay and Drew announce the poster awards, and then we're going to have a break until 4:30.
Okay. For the moment you've been waiting for, the poster awards. We have 5 categories, and we have an award for each of the categories. For the best foundational advance, Perseus Miscellaneous from the Holt Lab, and the title of the poster is Evaluation of Gene Expression from Large H. Influenza Genomic Segments Cloned in E. coli. Anybody here from the lab? Drew wanted to remind me that the prizes are being given by the BioBricks Foundation, and it's a nice check. Okay. Let's hope the next awardee is here. This is the problem with putting this off. Although they should have been staying for this session. For the best part, Chase Speisel from the Smolky Lab. A novel tool for programming cellular behavior, ligand-regulated RNAi substrates.
He had to leave early, but I can take his check.
Uh-huh. Make sure that isn't written out to Professor Smolky. If you go home with a new wardrobe. Best device. And the award goes to Samantha Sutton from the Undy Lab. And the title of her poster is Engineered Post-Translational Devices. Congratulations. Best application, Chris Anderson, the Voight Lab. Tumor-killing bacteria. Chris? The other Chris will come up and accept the award. And for best poster, Sierra Subramanian from the Weiss Lab. And the title of the poster is Engineered Cell-Cell Communication for Programmed Differentiation of Embryonic Stem Cells. Congratulations. Okay, Drew, do you want to come up and give us a few remarks about the day?
Well, first, thanks very much for what's been, for me, and I suspect for many, a tremendous day discussing and learning about many of the issues that, besides the technology itself and the excitement around its development and application, we're working to resolve. Coming from this meeting, we're going to have a written declaration, and as you might imagine, the exact words of this declaration do not exist at this instant in time. There will be a posting of a draft of the declaration, which will go online on Wednesday of this week, and we'll leave it online in draft form with comments to be received for a period of a week. Please send those comments directly to myself or to another email address that might be set up. We'll see exactly how that's put together. And then a week from this Wednesday, we'll release that. The purpose of our declaration is not, again, to revisit some of our conversations from the beginning of the morning, to recapitulate an Asilomar-like resolution for how we're governing ourselves, but rather to describe the process by which we're going to make progress on the issues that are facing the constructive development of synthetic biology. We all recognize the large number of possible positive applications that we'd like to go implement, and we need, in doing that, to directly address any possible concerns and optimize our organization to best let us pursue that. So if you have specific comments regarding any of the conversations from this afternoon, we might have a little bit of time to talk about it, or we could talk about it immediately after we close the full session. But we'll be working together with as much input as all input's welcome to pull together the draft. That'll, again, go online, and we'll freeze it a week from Wednesday. It'll be on the conference website. Every person attending the conference will have an email with that information.
That's open to, but that site is open to the world?
Yes. So there'll be a strong filter in place regarding, what comes in and whether it makes it into the revision. But we'll accept all input.
Okay, well, I'd like to thank you all for attending and for making this a really great conference. I'd like to thank the organizing committee, which was Drew, Chris Voigt, Adam Arkin, Wendell Lim. I'd really like to thank them for all their advice and input. And there are 5 people here who did all of the work for the last 3 days. They are Ellen and... Come on up. You can... Ellen and Randy and Grace and Kevin and Kristen. Why don't you guys come up to the front so we can thank you properly. Okay, so let's keep the momentum going and carry this forward to Synthetic Biology 3.0. I look forward to seeing you all there, and have a safe trip back home.